Okay, so hi everyone. So this is our fourth talk in the series, and this one is about using stable isotopes to measure biosynthetic rates. So the reason for this talk existing at this point is, so with our first three talks, we first of all did energy balance, did food intake, and then we started to move into more metabolic aspects with RER. And so in the next talk, I'm going to talk about how you do things like measure carbohydrate metabolism, lipid metabolism for phenotyping things like white adipose tissue. But a lot of those techniques become more powerful if you can use some form of tracer in the system, a stable isotope or a radioactive isotope. Now, the reason I got into doing anything with stable isotopes was we wanted to get a piece of equipment called a GC FID for measuring fatty acids. And for health and safety reasons, we were not allowed to buy one because it needs hydrogen. And so we had to buy a GC mass spec. And as a result of that, we had this shiny mass spec, which was about 4 times more expensive than a GC FID in the lab. And so began thinking, what can we do with it? So I'm going to talk through some of the things you can do with it. So in the talk, we're going to talk about what are stable isotopes and how do we measure them. I'm going to talk about some very specialized uses of stable isotopes, which are really the most basic ones, and that's what we got started with, but they're actually some of the most commonly used in animal metabolism. I would argue they're the most common ones of the simplest applications. We're then going to move to a more generalized example, which is deuterated water as a tool for measuring biosynthetic rates. And we're going to finish off with application of mass isotopomer distribution analysis for simultaneous assessment of product and substrate labeling. And hopefully by the end of this talk, you will understand what that sentence means. If you do now, that's great, but you probably know more than me. So what I would say then is, let's see how we go. Okay? All right, so what is a stable isotope? Okay, so you can have isotopes of elements, okay? So we're looking here at carbon. Carbon has 3 main isotopes. It has carbon-12, carbon-13, and carbon-14. They all have the same atomic number, as in 6, and they all have 6 protons, but they differ in terms of the number of neutrons they have. C14 will be well familiar with. C14 is vanishingly rare. It's less than 1 part per trillion, which for our purposes is effectively nothing. This is a radioactive isotope, and what this means is over time it will decay. In the case of C14, it actually turns into nitrogen because it emits beta radiation, which means electrons. So one of these neutrons emits an electron, becomes a protein. You now have 14N7 rather than 14C6. However, stable isotopes, and carbon has 2 stable isotopes, one is C12, one is C13. These do not decay, they are stable over time. And in the world around us, on average, we will find that 99% of carbon is the familiar carbon-12 of the periodic table, and about 1% is carbon-13. So carbon-13 is way, way, way more abundant than carbon-14. So how can we use the fact that we can have carbon that weighs 2 different weights? And so what we can do is we can buy molecules from our supplier, Goss Scientific, which are massively enriched in these stable isotopes. So you can have glucose that instead of having 1% carbon-13 in it, has 99+% carbon-13. Or you can buy deuterated water, D2O. And then when these molecules get used in synthetic reactions or they get broken down, and they can be incorporated into other molecules. And as such, any molecules that are newly made since we add our label, and we can add labels for really quite short periods of time, hours, minutes in some cases, if they're derived from our tracer, they will contain more of the stable isotope. And if we can measure that, we can get really accurate estimates of the rate of fluxes of things that are going on, rather than just looking at the steady state levels. So how do we measure them? Well, I would argue that nowadays the most common method for measuring stable isotopes is mass spectrometry. And there are many, many forms of mass spectrometer. I am not going to talk about them because I'm not an expert on them. Someone like Ben or Albert would be way better for that. But I will briefly explain about the one we use in our lab, which is a gas chromatography mass spectrometer. And it is one of the most basic that you can buy. And a GCMS consists of 3 main parts. It consists of a gas chromatography system to separate the molecules, an ion source to charge the molecules. Mass spectrometers all work by applying magnetic fields in order to separate things based on their mass charge ratio. So pretty much every mass spectrometer requires a charged molecule, and ions are charged molecules. So you need to ionize your molecules to be able to measure them. And then the mass spectrometer itself, which will comprise of a mass analyzer or a mass selector in most cases, and then a detector. Okay? So the first chunk is the gas chromatography system. So GC works like pretty much all forms of chromatography in that there are two phases. And so when we run an STS gel, for example, that's a form of chromatography. And we have two phases. We have a solid phase, which is the cross-linked acrylamide. And then we have the liquid phase, which is our running buffer. And the molecules that we study when we do a STS page gel are going to be proteins in most cases, and they will run through the gel based on their physical properties, and the physical property that most of the separation that is going on in a denatured STS page gel is going to be on mass. So bigger stuff runs slower because it's harder for it to get through these cross-linked things. With GC, we will have a stationary phase, which molecules bind to, and then a mobile phase, which is not a running buffer, it's gas. And the stationary phase, we can change that stationary phase to have different properties. So one major change is what the phase is made out of, so how polar it is. And usually you'll have sort of like an underlying backbone and then bits sticking out of it, which are going to be the polar groups. And so a common one is polysiloxane, and the amount of polysiloxane that's sticking out, you can adjust the percentage in your GC column, and that will affect what can run through it. So you might have 5% polysiloxane up to 50% polysiloxane. And again, going back to our analogy of a gel, an STS page gel, if you have a 4% gel versus a 12% gel, well, with a 4% gel, you know, all of your tiny molecules will just run straight through it and you'll never see them and you'll just get the big molecules. Whereas if you have a 12% gel, stuff over like, I don't know, 50 kilodaltons is not even necessarily going to get in it at all. So you're just going to separate small stuff. And so this is the same principle with the GC column. The gas phase is a carrier gas, and that moves the molecules along the column once they desorb from the stationary phase. And so the factor in this case, which determines whether they desorb, is temperature. So usually we'll start with a nice cold column, maybe about 40, 50 degrees. We'll inject our stuff and it will all stick at the start of the column within the first maybe inch or two of the column. And these columns are usually about 30 meters long. And then we ramp the temperature up. And as the temperature gets hotter and hotter, the molecules start to fall off and flow down the column. And then they come off at the end. And once they come off the end of our GC, they go into our ion source. So this is the interface between the GC and the mass spec. Here is the ion source. So how does this work? How do we get ions? So an ion source is basically an old school tungsten filament light bulb. So if those of you old enough to remember a time before the LED and halogen lights replaced all of these, old school tungsten filament light bulbs, you had a wire coil which was made of tungsten, and it got very, very hot. It was in nitrogen and it would give off light, but it also gives off tons of electrons. And so the type of source we're using is an electron ionization source. So our tungsten filament, what it does is it fires out electrons, and it does two things. The first is it breaks our molecule into fragments, and that pattern of fragments is a fingerprint because molecules will always break the same way. If you have the same energy for the ionization, they will always break the same way and form a fingerprint that identifies the molecule. And we'll come on to that in a second. And it also results in them becoming positively charged. And now we go into the mass spec. So as I mentioned briefly, there are two components of the mass spec. There's the bit that gives it specificity, which is which ions are allowed to flow. And there is also the detector, which simply detects how much stuff we have. So our mass spec is a quadrupole, and it works by having 4 magnets, hence quadrupole. The 4 magnets can be tuned to specifically allow ions of very specific masses through. So usually down to a window of 0.1 daltons. And the way it works is it does this, in our case, we're normally running it at about 4 times a second. So it would go from allowing ions of 50 daltons through to 450 Daltons through in 0.1 Dalton increments, so about 4,000 increments, and it will do that 4 times a second. And this is called scanning. So it scans up and down a range of frequencies. And at the end of the quadrupole, as I say, we have a detector, and that simply measures the amount of stuff hitting it. So, and the way it works is this: if you have tuned your quadrupole to 451.1 Daltons, and you have an ion of 451.1 Daltons, it is able to wibble through and hit the detector, generating a signal. If, on the other hand, you have now tuned, you scan up to 452 Daltons, it doesn't make it through, it crashes. And so you don't detect it. And so that's the principle behind how quadrupole works. Okay, what does data look like from a mass spec? It's basically a line. So what we have here is something called the total ion chromatogram. So I already mentioned we're usually scanning ions from 50 to about 450 Daltons. And what this does is it simply sums up over time all of those ions. So every second you're going to, or in fact it splits into quarter seconds because we do these 4 Hertz scans. So it's a splitting, these are minutes along the x-axis, and what we're doing is the total amount measured every quarter second gets plotted as a point. And so the more stuff we have, the higher the peak, the intensities on this side. And we're going to look at 3 specific molecules that we can see. Each of these peaks represents a separate molecule, and in this case, they're fatty acids. So they're fatty acid methyl esters, and essentially we're going to look at these 3, and these 3 are aladic, oleic, and becenic acid. And so these are all 18 carbon fatty acids, and they all have 1 double bond in them. Aladic fatty acid is a trans fatty acid, hence it's straight because this double bond is in a trans conformation. Oleic acid is C18-1N9, is a cis fatty acid, so it's got a kink in it because you have the cis conformation of the double bond. And that double bond is at the N9 position. This means it is 9 carbons from the methyl end of the molecule. Cisphosphenic acid is very similar to oleic acid, except this double bond is in a different location. It is 7 carbons from the methyl end and 11 carbons from the carboxyl end. So we've just moved the double bond. So you can see all of these molecules, all of these fatty acids, have exactly the same molecular weight because they have all got 18 carbons, they've all got 1 double bond, okay? And yet, despite their incredible similarity, we can separate them in a GC because of where the double bonds are located and also the conformation of the double bonds. So this is the total ion chromatogram, and now we've zoomed in just on cisphosphenic acid, oleic, and aladic acid, and you can see this is about 30 seconds of time. So these runs are quite long, they take about 30 minutes. And now what we can do is we can zoom in onto this peak, and we can actually ask, okay, so this is all the ions making up this peak, measured in total. What individual ions do we have in this peak? So if we zoom in, can we look at the mass spectra and look at how much of each individual ion there is? And indeed we can, and this is the mass spectra for cisphosphenic acid. And you can see these numbers here. This is like 55.1, 97.1. These are ions, and these are their molecular weight in Daltons. So this is a 55 Dalton ion. This is a 222 Dalton ion. But this one's particularly important, so I've put an arrow on top of it. This is 296 Daltons. And this is what is known as the parent ion or the full molecule. All of these other ions are fragments of the molecule because it's been smashed up by having electrons shot at it. But some of the intact molecule makes it through. So the intensity is on the y-axis, so how tall these peaks are is how much of that ion we have. And so why the parent ion is particularly useful for us is because it's very useful when we come to do any kind of stable isotope work. We know what the actual precise number of carbons, hydrogen, and oxygens in the molecule is, and you'll see why that matters later. So this ion here is 296, but we also have a 297 ion. Now this is actually the same molecule. It's still the same parent ion, but in this case, because we have naturally got 1% C13 in the environment, we're going to see about 20% of our oleate will contain 1C13. And the reason for this is, whilst it's only 1% in the environment, there are 18 possible locations that you can have a C13. So you're going to get 18 times as many chances to get a C13 into that molecule than if you just had a methane, which has 1 carbon, and then only 1% of methane will be C13 containing. And an important point is here, our 3 fatty acids we looked at earlier, elaidic, oleic, and cisphosphenic, their mass spectra are all the same. So because they just have 1 double bond, when the ionization process happens, the fragmentation occurs in the same way. And this is because in the high energy state, the double bond becomes unstable, the conformations can flip back and forth. And so in fact, our mass spectrometer has no power to distinguish between these 3 fatty acids. The power to distinguish between elaidic, oleic, and cisphosphenic lies in the chromatography. So we can separate them based on their physical properties, but when we ionize them and smash them up in our mass spec, oleic acid methyl ester and cisphosphenic acid methyl ester look identical. So the important thing to realize is that whilst it's easy to think the mass spec is the very powerful part of a GCMS or an LCMS, it's very often the chromatography on the front end, which is at least as important, if not more important, for accurately and specifically measuring what you want to measure. So, intro conclusion, GC separates our molecules based on their physical properties, e.g. their length, the number and position of their double bonds, and the cis-trans conformation of those double bonds. So this is for fatty acids. The mass spec tells us what the compound is, but only up to a point, because as we've seen already, it doesn't necessarily distinguish between molecules with the same molecular weight. The mass spec allows us to measure the abundance of specific ions, and natural stable isotopes mean we see multiple versions of the same molecules. So 296 ion will come along with 297 and 298 versions. These are called isotopomers, and we will mention terminology in a second. And if we therefore can add more of the stable isotope into our system as a tracer, we will see more of the 297, more of the 298 versions, and less of the 296 version of, for example, oleic acid. And so that's going to be the principle. And if we can quantify those things accurately, we can get some really cool biological information. Okay, we're going to do specialized uses of stable isotopes. This is part 1. Now, do not be scared. I'm going to put this slide up about 3 or 4 times because these are terms that get used when people are talking about stable isotope stuff. So, but I think it's good to just mention them once at the start, and we'll refresh on them as we go through the talk, okay? So a substrate, the substrate is the proximal thing that gets incorporated into your molecule, okay? The product is what gets made. M is a term you're going to see in reference to ions. So M and M+1, or M1, for example. And an ion made with the most common natural isotopes, i.e. it's all a fatty acid comprised exclusively of C12 and O16, is the M0 ion. So that would be the 296 that we were looking at for oleic acid. Or for palmitate, that's 270 Daltons. And the M+1, so when you see the term M+1, that means it's an isotopomer of 271 Daltons, and it contains 1 C13 or 1 deuterium if we've been using deuterated water. So isotopomer, M0, M1, M2, these are all isotopomers. N, big N, not small n, is the number of locations within the molecule that can be labeled. So if we're doing a study where we say use deuterated water, there are 33 hydrogens in a palmitate, and we'll come on to discuss the point, but there are only some that can be exchanged with water in the cell. P is the precursor enrichment. So this is the amount of the pool of substrates that is labeled. So a good example of this is a p-value with the deuterated water experiment. We do them in cell culture quite a bit at the moment, and we will use 8% deuterated water. So therefore our p-value is 8%. And F is the fractional synthesis rate. This is usually what you're trying to measure. So this is the proportion of the product that has been newly made during your experiment. So from putting your label in to harvesting your mouse, your cells, how much stuff got made is F, the fractional synthesis rate. Okay? Right. So measuring your labeled species. So this is just putting in a substrate and measuring a substrate. It's the first specialized case, and the second is tracing products with a one-to-one stoichiometry to the substrate. Okay, So why might you want to measure your label subspecies? Well, actually, we do this a lot in vivo. So one of the things we could do is for clamp studies. And so that if you just do a clamp without stable isotopes, you get a value called the glucose infusion rate. We're going to talk about clamping next time I do in the next talk. So I'm not going to go through it now. But if you add in a tracer, in addition to your GIR, you might get some values you may have heard of called the RD or RA, and the hepatic glucose production, HGP. That's dependent on having a tracer in the system. The other thing you can do is when you do GTTs, you can actually add in, instead of cold glucose that you get from like Sigma, what you can use instead is you can buy some stably labeled glucose, and then you can use that to split your GTT apart. I'm going to show that, and you can split it into the glucose you actually injected into the mouse and the glucose the mouse is producing itself. And so we actually published using this technique. So this was in a paper from that we published, I think, last year. And so what we did was we did a GTT and we found a difference in our animals. So the black were the wild types, white were the knockouts. So we had a somewhat improved glucose tolerance curve. But by using stable isotopes, we could split this curve into the exogenous glucose that we'd injected and the endogenous glucose that was basically coming from what was already in the mouse's blood, but also the stuff being produced by the liver. And actually, you can see in the wild types and the knockouts that the big difference is in the exogenous glucose levels. So it was showing us that there was altered clearance of glucose going on in the mouse, and actually there was really no difference in the endogenous levels. So it wasn't like the knockouts were demonstrating a big reduction in their own glucose production. They were essentially showing no difference from the knockouts. And interesting, you see a little spike here at 10 minutes in the wild types, and that's because if you pick up a mouse and stab it with a needle, which is what you're effectively doing with the GTT, no matter how careful you are, that will be a stress to the animal, and stress will cause glucose to go up. Okay, so the second specialized case is measuring one-to-one product substrate traces. And so the thing we most often do this for is to look at fatty acid remodeling. So we go, okay, we're going to put some palmitate on some cells, and we're going to ask how much of that palmitate gets desaturated, or how much of that palmitate gets elongated, so made into stearate, or made into palmitoleate, or made into oleate. So we're looking to measure the transformation of one molecule into another, and therefore we can measure enzymatic activities. The other thing you can do with this kind of approach is to track where stuff ends up. So we know that if we're putting palmitate into a system, we have loads of phospholipids, loads of different species. So we've got phosphoinositols, phosphatidylcholines, we have neutral lipids, triglycerides, we have sterile esters. So we can ask, if we put in a label under different conditions, where does our label go? Is it ending up in the neutral lipid, or is it ending up in the phospholipid? And this is the kind of thing we can do. So the kind of traces you can do are, you can either use the easy method, which is to use a highly isotopically distinct tracer. And so in an example of this would be U13C palmitate. So this is a palmitate where every single one of the 16 carbons in the palmitate has been replaced with U13C. And the advantage of this is it's very, very sensitive and it's very easy to analyze. The disadvantage is it's very expensive. And you can use a relatively isotopically similar tracer, e.g. 113C palmitate, and in this case, it's only 1 Dalton heavier than the normal naturally occurring palmitate. The advantage is it's cheap, but the disadvantage is you need to mass to deconvolute the natural isotopomers, because we know we get N+1 and N+2 ions, as we'll see in a second, because of naturally occurring 13 carbon in the environment. So here we go. This is a mass spectra showing palmitate. We have the parent ion, 270 Daltons here, and you can see here 271 and 272 and 273. These are the N+1, N+2, N+3 ions. If we use U13C, we see it out here at 286 Daltons. so 16 Daltons. And you can see there's absolutely nothing between about 273 and 286. So it's very easy to measure these things. You're not getting any overlap. And so if we did an experiment where we infused this U13C palmitate into a mouse for 3 hours at really, really, really high doses. And then we took the liver and we asked what had happened to the palmitate we'd infused. And we could see that within 3 hours, about 2.5% of the liver's lipidome was C16 palmitate that was labeled. Okay, and this was going, and you can see here, these are females in blue, males in gray, and we have vehicle controls in orange and yellow, and they have no label. And in addition to seeing palmitate in the phospholipids, we could also see C18 naught, which was M16 labeled. So this was the elongation product. So we'd see that maybe about 15 to 20% of the palmitate we put on in had been elongated to stearate. So we could trace the fates of where the stuff has gone. If we use 113C, it's a little bit trickier because this is our endogenous palmitate. And if we put 113C in the system, it will have the same mass as naturally occurring N+1 palmitate, 271. And so this creates this problem. If we have pure C13, this will be its spectra. So this is 113C palmitate. And if it's 100% labeled, it's going to look something like this, 99% labeled, in that you're going to have almost no M0, so you're going to have no 270 because every molecule has got one extra 13C, and the majority of it is going to be 271, 272, the M1, the M2 ions. Naturally occurring, you actually only have about 15% of it is this M1 ion. So if we observe our molecule and when we measure our palmitate, this is the ion pattern we observe, then this must be a mixture of molecules which are labeled and unlabeled natural molecules. So we need to deconvolute how much of this and how much of this as proportions will give us this. And to do so, we use some maths, which I'm not going to go into today, but essentially what we're doing is solving multiple linear equations using multiple linear regression. And you can do it actually in Excel. And what we can then do with this, again, this is another experiment, is tracing the fate of palmitate to multiple different products. And actually, this was an experiment we did in macrophages. And what we did was we put 200 micromoles of palmitate onto macrophages. And that will start to make macrophages very sick. And actually, what you find is part of the reason why it's making them sick is the macrophages can't deal with it. They start to have more and more and more palmitate building up in their membranes because they cannot detoxify it by desaturating or elongating it. And so you can see in these blue ones, which are wild type, you have 90% of that 200 micromolar palmitate you put on your macrophages stays as palmitate. And we could only track really about 3 or 4% of it being desaturated into palmitoleic acid, and maybe 7 or 8% of it being elongated into stearate. So the majority of the palmitate stays as palmitate when you're doing palmitate treatments on macrophages. So the applications of these approaches, we've mentioned them already. You can use them for tracer kinetic studies. As I say, we're going to cover this in more detail next week when we talk about clamps. And we can also pass complex experiments like we saw, like a GTT, into more components. So we can look at exogenous versus endogenous glucose. And it's really straightforward to do. And we can also measure lipid remodeling. So if we're specifically interested in the interconversion of a lipid, e.g. palmitate, we can use label palmitate as a substrate and look at its conversion to oleate. And so see how much that palmitate tracks through to oleate. Okay, so we're now going to move on to section 2, which is a more sort of generalized case. And this is a case where you can have multiple tracer molecules that get incorporated into one product. And deuterated water is a very common example of this as an approach. And in this case, we're going to know that our label can be incorporated at multiple sites. But really importantly, we're going to know how much of our substrate we have, how much of our label we have at the start. And so in order to know that, we need to be able to measure how much deuterium we have. And so one of the things that happens if you do a mouse experiment with deuterated water is if you put deuterated water into drinking water at about 4%, in body water, it's only going to be around 2% because a lot of water gets produced from respiration. So a lot of water comes from actually oxidizing food, and mouse probably is only obtaining about half of its water through drinking, and the other half is, as I say, coming from respiration. So you need to actually measure the concentration of the deuterium in the blood. Now, you can measure the amount of deuterium using isotope ratio mass spectrometers, but these tend to require really quite large amounts of serum. They're mostly set up for doing human work, so I'm not going to talk about those. But you do not often get 400 microliters of serum from a mouse that you can readily use. So I'm going to talk about a method which we got from Stephen Previs's lab. He was very helpful with it, and this reference here I'm just going to mention to you because it's really good. And the way it works is by an indirect way of measuring deuterium. Because you cannot measure water readily in the mass spectrometer and through the GC, but what you can do is you can exchange the deuterium from water onto something else that we can measure. So this is deuterium. Just to fill you in, this is a hydrogen. It has 1 proton, 1 electron. Deuterium, stable isotope, 1 proton, 1 neutron, and 1 electron. And deuterated water, D2O. So as I mentioned, we're going to exchange our deuterium from our water, which we can't measure, onto a molecule we can measure. And the molecule we're going to use is acetone. And acetone is great for this purpose because it has uniform hydrogens. So every hydrogen is bonded to a carbon that's then bonded to a ketone group. It's miscible with water, so we can mix it with our aqueous, either cell culture media, serum, or deuterated water itself for making a standard curve. And it partitions really well into the gas phase, as anyone who's opened a bottle of nail varnish remover will know, because you can smell it really strongly. And so this makes it perfect. And what we do is we catalyze with sodium hydroxide the exchange of the deuterium from the hydroxyl ions of water with the hydrogens of the acetone. And so this actually happens pretty quickly because what we're going to use as a catalyst is 10 mole of sodium hydroxide. So we take only 5 microliters of serum. We then take 4 microliters of sodium hydroxide and 2 microliters of acetone, mix them together, wait 4 hours, and then we do something called headspace sampling. So we don't actually inject the liquid into our mass spectrometer. We just inject the gas above the liquid. And that's going to contain, obviously, air as the main component. But because acetone partitions into the gas phase very readily, it's going to contain a lot of acetone. And so here's our GCMS trace. We previously saw 37 fatty acids. Here we see 2 big peaks. One is air and one is acetone. And if we look at the chromatograms of acetone that has been reacted with sodium hydroxide and water or deuterium or deuterated water for 4 hours in the presence of 10 mole of sodium hydroxide, you can see here, acetone has a molecular weight of 58 Daltons, and you really don't get much fragmentation of acetone in the mass spec. So at 0% deuterated water, you have about 97% of it is 58, and because we've got 3 carbons in an acetone, about 3% of it is 59. If you have reacted with 100% deuterated water, you will see that 58, 59, 60, 61, 62, 63, and 64 are all present. So we have some acetone which has all 6 of its hydrogens having been replaced with deuterium, and the majority of it carries either 2 or 3 deuteriums. And then all you do is you create a standard curve comparing the 59 to 58 ratio, plot this out, and then any sample you have, you can analyze the amount of deuterium in it. Okay, so how do we assess deuterium incorporation into newly synthesized product molecules? So we now know how to measure how much deuterium we have, which is one of the pieces of information we need. We now need to work out how we take the information from measuring our molecule of interest. Okay, so just going back to these abbreviations, and the ones to really bear in mind now are the N value, the number of locations within a molecule. We're going to go through that. P, the precursor enrichment, so this is the amount of pool of substrates that's labeled. In our case, that's our deuterium concentration for this section. And F, the fractional synthesis rate, which is what we are going to be interested in. Okay. So when we have a tracer that's capable of labeling multiple positions in a molecule, it is critical we know how much of the precursor pool is labeled and how many locations the precursor can be incorporated into the molecule, because both of these will affect the ultimate ion pattern of our product, the isotope distribution. And we can think of this like a car park to explain why that's the case. If we have 6 places in our molecule, so like acetone, that can be labeled, and our precursor pool is labeled at about 16%, then we would expect there to be about 1 label introduced per product. So this will give us the N+1 ion. If we increase the precursor pool labeling to 33%, then we would actually expect in general to get some more of the M2 ion. That will be the predominant ion, as the most likely number of labeled in this case, say deuteriums within our acetone would be 2. Conversely, if there are 12 places in our product molecule that can be labeled and our precursor pool is back down to 16%, then we would expect there to be about 2 labels per product, and this gives us an N+2 ion. Now, obviously, the math is not as simple as just multiplying A by B, but this is a sort of a general idea. So you would get an N+2 ion in this setting with 16% P and an N of 12, and you get your most likely one to get is an N+2 ion if you have an N of 6 places, and 33% P. So they both affect. So if we know how many car parking spaces we have and the labeling of our precursor pool, we can easily calculate the ion distribution of our newly synthesized molecule. And this is handily for D2O. Many of the N, capital N, number of car parking spaces, values for commonly synthesized molecules have been calculated. In our case, the one we're going to look at is palmitate, and that is, if you go to literature, that is 22. That's in vivo, and we've done the experiments ourselves. It's absolutely rock solidly 22 in vivo in our hands. There is a lot of literature, and we've found this, that it varies in vitro, which is quite interesting. It seems to be due to a catalysis of the exchange between water and NADPH by flavoproteins, but I'm not going to go into that. There are papers about it. But as a general question, it's quite interesting that although palmitate has an N of 22, it actually has 33 hydrogen in it. So why in vivo is very defined as 22 is the N number, why the other 11 are inaccessible, I think it's still not necessarily 100% clear. So now we can see the effect of changing our P number. So this is sort of the simple idea of just increasing the amount of precursor pool. And so what this is going to show is the natural ion of a normal palmitate. So that's got an M0 of about 82%, M1 is around about 15%, M2 is down about 2 or 3%. And then if we imagine a entirely newly synthesized molecule of palmitate that's been made whilst we had 0.5% deuterated water in the system. And so you can see here then, this has some effect on the M0, drops it by about 5 or 6%, bit of an increase in the M1 and a bit of an increase in the M2. Probably this is the largest change, it's about double. But it's really quite noticeable here that they're not that different, these molecules, to look at, okay? If we increase to 1% deuterated water, you can see that they start to get quite a bit more different. So M1 is now nearly double, M2 is probably about 3 or 4 times, and M3 is starting to become quite measurable, 2%, and then 3%. So you're getting really different ion spectra. It is easier to deconvolute, that deconvolution we did earlier to separate our palmitate that was 113C from our naturally occurring. It is easier to deconvolute mathematically how much of the blue molecules you have and how much of the orange molecules you have if they're more different. So higher percentages of labeling are helpful in this circumstance. Equally, we can look at the effect of N. So if we look at 2% deuterated water, this is would be what happens with an N of 11. That's with an N of 22. and that's with an N of 33. So a fixed level of precursor, we're just changing the number of car parking spaces. And this is showing like the calculated distributions. Now, the important point here is if we are doing one of these experiments, we only have two types of molecule in most likelihood. And the two types of molecule we're going to have is orange molecules or blue molecules. So we should not be getting too many intermediary species in a setting where we're doing, say, DNL. And so this is very useful because then we can separate how much of the orange molecules and how much of the blue molecules we have. And it's the same thing we did earlier with the 113C palmitate to separate the 113C signal from the natural signal. And so we can use the math system, or we can simply use the following equation, which is the one quoted in the literature, which is product enrichment. So all you do is you add up M1, how much more of the M1 you have, so like the orange, than you would expect here, subtract that from that, multiply it by 1, grays, subtract the observed from the natural, multiply it by 2, yellows, subtract this from the natural, multiply it by 3, and then if you know your N, 22, and you know your P, because you've measured it in the case of our deuterium, you can just calculate it out. And both methods work very, very reliably. We've updated this now. It's an extremely good correlation, and they basically give the same result. Okay, so what can we apply deuterated water to? So the measurement of fatty acid synthesis, certainly, the measurement of cholesterol synthesis, the measurement of amino acid synthesis and protein synthesis can potentially be done, glycogen synthesis. If you use pulse chase designs, you can look at degradation and turnover rates, say of lipid and adipose tissue. And this is really, really powerful, but it is important to considering the labeling time, and we'll come to that in 2 slides, as due to the issues of steady state, and also you need to consider your deuterated water concentration to give you the optimal separation when you're doing mass spec. Okay, and well, in my opinion, you can get some pretty amazing results. So this result actually like blew me away because I did a labeling experiment with deuterated water, and I decided to measure how much palmitate gets biosynthesized each day in the livers of mice. And so I put in deuterated water, and this is the fraction of newly synthesized palmitate, not the percentage. In other words, in the neutral lipid fraction, so things like triglycerides and cholesterol ester, in female and male mice, this is on a chow diet, so it's got 20% fat in it. About 45% of all the palmitate was newly synthesized each day. This is 24 hours. In the phospholipid fraction, that actually rises, and that was around 57, 58%. And I assumed I had done this wrong, so I went trawling through the literature, and I found a study from 1950 that had done this with radiation, and they report a half-life for palmitate in the livers of mice of about 24 hours, which fits pretty well with an average biosynthetic rate of 50% in 24 hours. So this is half of all palmitate in the liver of your mouse wasn't the same palmitate as yesterday, which I think is quite amazing because I would not have thought it was that high, even in rodents with their super high metabolic rate. And actually, though, this highlights a problem because this we're measuring the fraction of newly synthesized palmitate, and it's appearing to be 58%. It's probably higher. And it's probably higher because of this problem. And it is as you label more and more of your stuff. So this is how much product is labeled in your sample. And it's going from 0 to everything being labeled, and the y-axis is time. As you label more and more of your stuff, so we can come up here and say, okay, 58% here, it's taking about 36 hours. You start to replace newly synthesized molecules with newly synthesized molecules. And so, in fact, by the time you're getting up to 80% of your molecule has been labeled, to get to 81%, you're going to need to do 5% more synthesis because you're going to replace lots and lots of newly labeled stuff. And you can only distinguish between newly and old. You can't distinguish between newly labeled and subsequently newly labeled again. And so what happens is, therefore, your apparent rate constant, how fast things are happening, falls as a function of how much has got labeled. So we're estimating 58% is newly synthesized. In the phospholipids, is probably even higher. Okay, final section. So this is mass isotope distribution analysis. This is a technique which allows us to measure the levels of P, the product enrichment, and the fractional synthesis rate without knowing P. So with the deuterated water experiment, we measured P because we measured how much deuterium was in our sample. And then we used that to calculate our expected distribution of the ion pattern, and then we used the mass to calculate the fractional synthesis rate. So to do this, we're actually just going to use the isotope distribution of, in our case, palmitate, to calculate P, i.e. our product enrichment, as well as our fractional synthesis rate simultaneously. And there is good reason that we may need to do this. So I'm just flashing this up if anyone wants to refresh themselves on these again. I'll give a couple of seconds, but I'm not going to go through them again this time. Okay, so why might we want to do this? Well, the reason we might want to do this is if we actually want to measure something that is intermediate between our ultimate product and the tracer we put on the cells. And so this is an experiment from my colleague Guillaume, and we did this together probably about a year ago now. And what we were trying to do was the following, and we were trying to trace how much NADPH was getting consumed in macrophages, in this case, by de novo lipogenesis. Okay? I'll explain why later on we're doing that. But the way we were doing this was we were using glucose, which had a deuterium on its 3-carbon. So the 3-carbon has a hydrogen and an OH group on it. The hydrogen on it was replaced with a deuterium. And the reason why this is useful to us is because this enzyme, 6-PGD, which is part of the pentose phosphate pathway, when this enzyme does the interconversion of 6-PG to ribulose 5-phosphate, it removes this deuterium from the 3 position, and it puts it onto an NADP plus to produce an NADPH. Okay? So we could put on loads of this labeled glucose, but the NADPH has multiple cellular sources, and the P we need to know is not how much, what the concentration of the glucose was in the cell culture media. It was actually what proportion of this NADPH is labeled. And in concept, and we did try this, you can do it with a very, very expensive mass spectrometer, but NADPH is a really difficult molecule to measure. It's also a high energy molecule, which makes it, you've got to extract it very, very carefully. It's very hard to do. It's prone to fall apart. So actually what we ended up doing was to calculate the enrichment of the NADPH pool, as well as the fractional synthesis rate of palmitate, by just measuring the isotope distribution of palmitate. I'm going to show you how we did it. And first of all, I'm going to tell you why we wanted to do it. And so the reason we were doing this, first of all, was to actually accurately quantify fatty acid synthesis. So Guillaume had a model for how DNL affects macrophage polarization in response to IL-4. And the argument is that IL-4 actually affects polarization by modulating ROS levels, and it does so by activating AKT and SREBP1. This increases fatty acid synthesis, and fatty acid synthesis consumes NADPH. NADPH is not just used for fatty acid synthesis, it's also used for redox defense. So it regenerates GSH. If you use all your NADPH for fatty acid synthesis, you can't make enough GSH, and this means ROS increases. And actually, in this case, ROS was not a bad actor, but in fact is a second messenger promoting alternative activation. So if you block fatty acid synthesis in response to IL-4, then you don't get polarization to M2 polarization. Okay, so the issue was at the time, what we had was a radioactivity experiment. And it was expressed in fold change. And so we had control here is 1, IL-4 here is plus, and that would boost the amount of fatty acid synthesis by 1.4-fold. And actually, if you block AKT, which is here, then that would reduce DNL, not surprisingly, because fatty acid synthesis is downstream of AKT. It would reduce it down to about 70% if you blocked AKT. So 1.4-fold. But 1.4-fold isn't much use, actually, if that's just molar percentage. So it's 1% of the fatty acids got made, and that went up to 1.4. Because would that actually be a biologically meaningful amount of DNL in order to reduce this NADPH level and impact on GSH? I think it would be problematic. Particularly given IL-4 didn't hugely increase the amount of DNL apparently going on in the cell. We see a slight, the darker bars here, increase in palmitate, maybe 1 to 2%. But what we also see is a slight reduction in stearate. So it might just be a change in elongation. And if you think about the example I showed you earlier, though, just because you don't see a big change in the amount of lipid doesn't mean there's not a lot of lipid synthesis going on. Let's think about our mouse livers. They're not gaining 50% liver lipid weight every single day. This is a turnover. So it's possible we weren't seeing a big increase in the amount of palmitate in the cells, but we might be seeing a lot of turnover. Okay, so we go back to this system. So this was what we were going to do, 3,3-H glucose, calculate both our NADPH pool and also measure the amount of palmitate synthesized over a period of time. Okay, so how can we do this? So we're trying to get two things. We're trying to get the P and we're trying to get the fractional synthesis rate. And actually, it turns out that the relationship between the peak heights of ions, and in particular the ions M2, M3, and M4, is dependent on your p-value, regardless of how much fractional synthesis is going on. Okay, so you can see here we've got some theoretical p-values, and I've drawn out the M2 to M3 and M4 relationships with arrows, and I'm just going to compare the 20 in orange and the 9 in gray. So you can see if you have a p-value of 20, your M3 and your M2 and your M4 are going to be largely the same height. If, on the other hand, you have a p-value of 9, your M2 is going to be twice as big as your M3, and your M3 is going to be twice as big as your M4. Okay, and there's very little, particularly in M3 and M4, naturally occurring isotopes at this molecular weight. So it's pretty much all down to that p-value effect. So when we looked at the actual data we got off the mass spec for palmitate, it actually had this sort of halving and halving again relationship, which was really more indicative of the red line here, which is around about 9%, rather than 20 or 4%. The amount of the distributions is really dependent on F, and this is definitely strongly reflected in how much M0 is dropping and how much M1 is increasing. And so from this data, you can then set up an equation system where you will solve both P and you will solve F simultaneously. And this is what we found. That 1.4-fold change was actually 20 to 28% of the cellular palmitate in this first experiment. I think that was over 24 hours, but Guillaume can remind me. But I think it's actually, we're pretty much now down to 40 and 60% has been the values we obtain with deuterated water. Because I think the conditions have been changed to do this over a shorter period of time because we overlabeled. So what you can see here is the treatment actually is increasing the lipid biosynthesis. This is all the palmitate in the cell by about 8% per day. So that's a pretty decent chunk. And what we're actually able then also to do is to look at the fraction of labeled NADPH and also measure the concentration of NADPH and therefore calculate how much NADPH is being turned over in the same time period. And we can also calculate from the palmitate biosynthesis how much NADPH is being consumed. So we could actually work out we were consuming sane amounts of NADPH to be a meaningful regulator of redox homeostasis. And this is one of the cool results from this. IL-4 didn't just increase de novo lipogenesis, how much palmitate was being made, it actually altered the cellular source of the NADPH. So the fraction of NADPH, the p-value that was labeled, was increased by IL-4 from about 6% to about 8%, highly significant response to IL-4. And the reason this can happen is because there are multiple sources of NADPH production. The one we were looking at is this enzyme here, part of the pentose phosphate pathway. And in vivo, in liver, we think this is really the major form. In cells, lots of other processes, including IDH enzymes, GLUT, which is involved in the entry of glutamate into the citric acid cycle, but also in particular malic enzyme. And this is thought to be a very important source in both macrophages and 3T301 adipocytes of NADPH production. So malic enzyme. So it is entirely possible that if the balance of which enzymes are responsible for NADPH production changes, we can see a change in our p-value, because if we start doing more pentose phosphate pathway in response to IL-4, more of this, and less of this, our label on our 3,3D glucose only can enter into the NADPH pool at this enzyme. And what's really nice is we had RNA-seq data from this, and we looked at the ratio between 6-PGD and ME1. And what we find is the fold change in PGD divided by the fold change in ME1 is about 1.4-fold increased in treated. In other words, suggesting that the PPP pathway, at least on the transcription level, is being favored over the ME1 pathway when you treat cells with IL-4. And this compares to the fold change in the NADPH as a portion of the pool. Okay. So in summary, at this point, stable isotopic tracers can be used in vitro and in vivo to probe different aspects of metabolism. We focus a lot on biosynthesis in this. And when you combine them with analytical techniques such as solid phase extraction, thin layer chromatography to separate your molecules, and gas chromatography, followed by mass spec, it's possible to trace almost anything. So almost anything that will incorporate carbon or incorporate hydrogen, and you're looking at most stuff there, or nitrogen, and you're looking at pretty much everything, can be traced in some way to measure some aspect. And really the only limitation is your imagination and what you want to measure.